Good afternoon, folks. Hope everybody is doing well. This is starting up part 3 of my lecture series on the fundamentals of the Kingdom Animalia. So if you are a member of my bio class and you have access to Blackboard, you might want to make sure that you have the summary notes that will have some important vocabulary terms to follow along with as you listen to this lesson. It's the same one that you used for parts 1 and 2. So we talked now about and by the way, yeah, if you're just coming on, if you have not yet watched part 1 and part 2 of Fundamentals of Animals on my channel here, you should go back and make sure you do that before you continue so that you have important background information that's going to be needed for this section. All right, so we've taken a look at what it means to be an animal. We have talked about the distinguishing features of Kingdom Animalia, the evolution of animals from earlier protist precursors, and the history of the Kingdom Animalia from the beginnings of simple animals up till the present era. So what I want to talk about now is a very important feature for discussing animals, and that is looking at the animal body plan, because more so than any other kind of life, animals have very sophisticated body plans, and this is key to the classification of animal diversity, key to our understanding of how we categorize animals and see how they fit into their environments. Now, when we talk about a body plan, this refers to a set of common morphological and developmental traits seen in a given animal species. A set of common morphological and developmental traits seen in a given animal species. One of the important features of being an animal is a high degree of morphological consistency, that you can look at two animals of the same species, and in general, you will find that they resemble one another more than they do things outside of their species. There are some weird exceptions. There are some species with very high degrees of morphological diversity. Think about the high average heterozygosity of dogs and of fruit flies, for example. But in general, most animal species have a fairly consistent body plan by which we can identify the animal and say, yep, those features tell us what kind of animal we are dealing with here. That is a hammerhead shark, that is a gray squirrel, whatever else it happens to be. The body plan reflects the adaptations of the animal to its particular environments. This is a way that we can understand not only taxonomy of animals, but how they have evolved and adapted to fit into their particular environments. Now, at one time, we looked at animals and we categorized them in ranks of complexity based on the sophistication of their body plan. Aristotle's Scala Naturae from 2,500 years ago is looking pretty much at that, looking at the arrangement of animals in a hierarchy of perceived complexity. And although he was talking in terms of vital heat, that was his basis for classification, if you look at the animals, you'll find that he's basically looking from simple animals to complex animals. Now, we know today that evolution is not hierarchical, nor is it progressive, that you don't look at a complex animal and say that animal is more evolved than a simple animal. But it cannot be denied that some animals have simpler body plans than others. This does not mean they have evolved less or evolved more poorly, because they're still well suited to whatever environments they happen to occupy, and many of them have been in those forms for millions of years. Keep in mind that if an animal hasn't changed much for a very long time, that tells us primarily that its body plan is working pretty well, and therefore is not an evidence that it is less evolved or anything like that. You can't have more or less of evolution. So we can talk, pardon me, allergies. We can talk about grades of complexity in the animal kingdom. That two animals are considered to be of the same grade, you might say, if they are approximately of equal complexity, regardless of whether or not they are closely related. Don't confuse a grade with clade. The term clades, C-L-A-D-E, refers to any group of related organisms. That's a clade. Whereas a grade is, any level of complexity, which does not necessarily imply close relationship. Sea slugs and land slugs are not closely related to each other, but nonetheless, they are at about the same grade of anatomical complexity. So, how do we talk about animal body plans? How do we classify the complexity of animals? A few different ways we can discuss this. First and foremost, let's talk about a thing called symmetry. Now, symmetry is the consistent repetition of parts in a stable pattern within the body plan of a given animal. Now, there are a couple of forms of symmetry we can discuss. I should point out first off that the very simplest animals, the sponges, are considered asymmetrical. Symmetry is S-Y-M-M-E-T-R-Y. If an animal is asymmetrical, it's A-S-Y-M-M-E-T-R-I-C-A-L. Asymmetrical. Sponges are asymmetrical, prefix A without. So sponges lack symmetry. They do not have a consistent repetition of body parts around any kind of axis or plan. A sponge is not guaranteed to look the same on the left as it is on the right. Sponges lack that, and the very most primitive animals were probably all asymmetrical like them. Now, in nature, there is a form of symmetry that animals do not exhibit, and that is spherical symmetry. If an organism has spherical symmetry, that means that it is symmetrical in 3 dimensions around a central point. In other words, the animal has no distinct top, bottom, left, or right. Its body plan is a sphere. We find spherical symmetry primarily in colonial protists, like volvox. There is no living animal that exhibits spherical symmetry. Some of the early proto-animals evolved from colonial protists may have had spherical symmetry, but we have nothing like them now. In living animals that exhibit symmetry, there are 2 basic forms of symmetry. We can talk about, first off, radial symmetry, R-A-D-I-A-L. Radial symmetry is repetition of parts in a circle around a central axis. In other words, a radially symmetrical animal does not have a distinct left and right or a distinct front and back. It is the same all around the body in a circular pattern around a central vertical axis. Your radially symmetrical animal will have a distinct top and bottom, but it will not have a distinct front back or a distinct left right. Some radially symmetrical animals, I should point out, do have body parts that are only found on one side of them, in which case you might say, okay, that's the front, but there still is no lateralization of left versus right parts. Radially symmetrical animals are very simple in their orientation. Lacking distinct directionality in your organization makes it hard to navigate in a terrestrial environment, and therefore it should come as no surprise that all radially symmetrical animals are those that live in marine or aquatic environments. They are very simple body plans. They are low-grade animals in the sense of not exhibiting a lot of structural complexity. We find radial symmetry only in the very simplest of animals, primarily in the phylum Cnidaria. That's capital C, lowercase n-i-d-a-r-i-a, Cnidaria. That is the phylum containing the anemones, the jellies, like your jellyfish, the hydras, the very simple, many cases, sessile animals with very sac-like body plans organized around, again, that central vertical axis. There are a couple of other simple animals that exhibit radial symmetry. Certain members of the phylum Ctenophora, capital C, T-E-N-O-P-H-O-R-A. That is the so-called comb jellies, another jelly-like organism, exhibit radial symmetry. Starfish, sea stars, or members of the phylum Echinodermata, E-C-H-I-N-O-D-E-R-M-T-A-T-A. No, M-A-T-A, my bad, Echinodermata. The echinoderms, like the sea stars and the sand dollars, also exhibit radial symmetry in the adult form. Interesting thing about the sea star is that members of the phylum Echinodermata are nifty because they often exhibit bilateral symmetry in larval condition, but then radial symmetry in the adult condition. So that's kind of a nifty thing. Now that brings us directly to our much more common and more complex form of symmetry, and that is bilateral symmetry. B-I-L-A-T-E-R-A-L. Lateral meaning to the sides, bi means two, bilateral, two-sided. In other words, if you have bilateral symmetry, you exhibit symmetry down the left versus rights. That basically one side of the body matches the other side of the body. And so bilateral symmetry, two-sided, your body is mirror imaged around left versus rights. Bilaterally symmetrical animals have a distinct left and right, a distinct front and back, and a distinct top and bottom. Most living animals exhibit bilateral symmetry, and all terrestrial animals exhibit bilateral symmetry. The majority of bilaterally symmetrical animals also exhibit what's called cephalization. C-E-P-H-A-L-I-Z-A-T-I-O-N. Cephalization. Prefix C-E-P-H. Ceph means head. Cephalization is the development of an anterior head, having a head at the front of the body, the head being a distinctive anatomical structure containing specialized sensory organs and generally containing a high concentration of nervous tissue. Basically, if you have a brain or equivalent structure, the head is where it's going to be located. Most bilaterally symmetrical animals exhibit anterior cephalization. This is a feature that's quite useful when you're navigating in a terrestrial environment. A bilateral animal has a body plan much more suited for moving around in a 2D world, that is a flat world, as opposed to the 3D world of the aquatic environment in which the radial symmetry works pretty well. But bilateral symmetry is about the only plan that meaningfully works in a terrestrial environment, and that basically requires having a forward orientation for the body and therefore placing important sensory receptors at the quote-unquote front, hence the development of a head as a useful evolutionary feature. I should point out that radial symmetry is the older and more primitive of the two. The most simple animals that are not sponges all exhibit radial symmetry. And then you're going to have your bilateral symmetry as a later evolutionary development. As far as we can tell, all bilaterally symmetrical animals share a common bilateral ancestor. In other words, bilateral symmetry is a monophyletic quality. Pardon me. Whereas radial symmetry seems to have appeared independently in several distinct phyla of life. So symmetry is one important way of talking about the body plan and how it's organized. You can see why this is a very fundamental organizational feature for your animal. Beyond that, let's talk about some other things that are important parts of the animal body plan. Let's look at tissue development. Now, remember, not all animals exhibit tissues. Sponges, as our simple basal animals, do not. But everything that isn't a sponge has distinctive tissues. And tissues are groups of cells that arise from the same region of the embryo and perform a common function. Sponges lack them. All other animals have them. Now, your tissues begin to appear early in embryonic development. Following gastrulation, remember gastrulation, where the developing blastula begins to pinch and fold in on itself, and you get this multi-layered sphere with a hollow interior. After gastrulation, now that you've got this multi-layered ball of cells, the distinct layers begin to sort and organize themselves into what we call primary germ layers, sometimes called embryonic tissues. There are three basic kinds of embryonic tissue that develop in animals. Some animals get two of them, some get all three. All animals early in their development will develop a primary germ layer on the outer surface of the gastrula called the ectoderm. Prefix ecto, E-C-T-O, suffix derm, D-E-R-M. Ecto means on the outside of, derm means skin or barrier. So outer skin. The ectoderm is going to form the outer coverings of the animal's body, and in many species, it will give rise to the central nervous system. If you go through a nerula stage of development in which a neural streak appears, it appears in the ectoderm. Now, you're also going to have an inner layer, a lining of that hollow sphere that we formed during gastrulation. The inner germ layer is called the endoderm. E-N-D-O-D-E-R-M. Endo, of course, you all know means within or inside of. The endoderm, or the inner layer, is going to give rise to the digestive tube. It's going to give rise to numerous internal organs like lungs and livers. From the endoderm, we will produce the primitive digestive tract, which is called the archenteron. Archenteron. The archenteron is an old term for the basic digestive tract, the continuous tube that runs from mouth to anus. And that's going to arise from the endoderm. Now, if I am an animal with only two primary germ layers, endoderm and ectoderm, I am said to be diploblastic. Diploblastic, or I am a diploblast. Diploblasts are animals that only have those two basic layers. Only very simple animals are diploblastic. We are going to see this condition primarily in our cnidarians and their affiliates, jellies, corals, things like that. Most animals are going to be triploblastic. Triploblastic. Triploblastic, or triploblasts. Triploblasts develop a third primary germ layer between the ectoderm and the endoderm. And that third layer is referred to as the mesoderm. The prefix MSEO, meso, means in the middle of. So the mesoderm is going to form in our triploblastic animals. It's going to give rise to muscles. In animals that have a skeleton, it's going to give rise to the bone, and it's going to give rise to any organ that would otherwise be found between endoderm and ectoderm in the animal's body. All bilaterally symmetrical animals are triploblastic. So if you are a bilaterian, that is an animal with bilateral symmetry, you are also a triploblast. So whereas most of the things that are not bilateral are going to be diploblasts. All right, so that's another way we can understand animal body plans in terms of the germ layers that it develops. You get 2 or 3. The more complex tissues are going to arise from those primary germ layers as the organism makes the transition from gastrula to embryo. And finally, another aspect of animal body plan is going to be body cavities, the spaces within the body. Now, if you are a triploblast, you will very frequently have what is called a coelom. Now, the spelling of that word does not sound like what I just pronounced. Coelom is spelled C-O-E-L-O-M, coelom. A coelom is a fluid-filled body cavity that arises from the mesoderm. Most triploblasts are going to have a coelom. If you have a coelom, you are said to be a coelomates. C-O-E-L-O-M-A-T-E-S. You are a coelomates if you have a coelom derived from the mesoderm. Most triploblasts are coelomates. We are coelomates. We are, of course, we're triploblasts, we're bilaterians. That's all implied in there. Now, some animals have a coelom that is instead derived from the blastocele, that initial compartment that forms inside of the blastula, that fluid-filled compartment. Sometimes that is the source of the coelom. If an animal derives its coelom from the blastocele, it is said to be a pseudocoelomates. 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 And I am looking at the handout I printed for you guys, and I think there's a spelling error in there. I think pseudocoelomate was spelled wrong with the E before the O. Check that and correct that if that is the case. So a pseudocoelomate, prefix pseudo means false, false coelom. Pseudocoelomates derive their coelom from the blastocele rather than from the mesoderm. Nematodes, roundworms, are a good example of a pseudocoelomates. And of course, there are some animals that lack a coelom altogether. They do not have a fluid-filled internal body cavity. If you lack a coelom altogether, you are said to be an acoelomates. A-C-O-E-L-O-M-A-T-E. Prefix A means without. Flatworms, for example, are acoelomates. Jellies are acoelomates. They have a central body cavity, but it's not a coelom. It's not a special fluid-filled compartment. It is rather a just an opening into their sac-like body plan. Sponges, I should point out here, fun thing about sponges. Sponges are not coelomates, they are acoelomates, but they do have an internal body cavity, which is sometimes called the spongocoel. S-P-O-N-G-O-C-O-E-L. The spongocoel is the space inside the soft porous body of the sponge, where you're going to find the collar cells that capture its food, and the reproductive cells that give rise to its eggs and sperm. Now, if you've got a fluid-filled body cavity, if you have a coelom, whether it's pseudocoelomate or proper coelomates, fluid-filled body cavity gives you some special advantages. First off, it cushions and protects your internal organs, separates them from the body wall so that they're not pressed up against it and therefore more subject to external damage. Furthermore, if you lack a bony skeleton, your fluid-filled coelom can provide a source of mechanical resistance for purposes of muscular action. Muscles need something to act upon. They need a source of resistance in order to create movement. In your body, we have an endoskeleton, E-N-D-O, skeleton, S-K-E-L-E-T-O-N, endoskeleton. That means you have an internal bony skeleton. That's the framework for your muscles that provides them with a source of mechanical resistance to pull upon, to generate your movement. A lot of invertebrates are going to use their coelom as a source of mechanical resistance. That muscular action is going to act on fluid-filled compartments. If you do not know this, water is a non-compressible fluid. You cannot squish it into a smaller area, and as such, the coelom can serve as a source of mechanical resistance for locomotion. We call that a hydroskeleton, H-Y-D-R-O, skeleton. All right, and last but not least, we're talking about body plans. Let's talk about the development of the body plan. There are a couple of basic developmental modes that we see in animals, and the body plan comes out in different ways depending on which developmental mode you have got. Two basic developmental patterns that all animals exhibit one or the other. Animals can be protostomes, P-R-O-T-O-S-T-O-M-E, or we would say they exhibit protostome developments, or they can be deuterostomes, D-E-U-T-E-R-O-S-T-O-M-E. And if you want to do a comparison, contrast, a set of columns side by side to lay this out in your notes and look at the features of them, this is not a bad idea. So you either exhibit protostome development or deuterostome development. And these are basically developmental patterns of how the body forms in its early embryonic developments. First off, how do cells form? Protostomes exhibit what is called spiral cleavage. Contrary to the name, spiral cleavage is not a trick that Candy does down at the strip club. Spiral cleavage is a form of cell division in which you see the formation of new cells in a diagonal or spiral fashion around the central axis of the animal's body. In other words, we lay down new cell layers diagonally spiraling around the central axis of the embryo. In addition, you're going to find that your protostomes exhibit determinate cleavage. D-E-T-E-R-M-I-N-A-T-E, determinate cleavage. In determinate cleavage, individual cells assume specific cellular identities early in development. In other words, determinate cleavage is characterized by early differentiation of embryonic stem cells, meaning that those cells don't stay embryonic stem cells for long. They very quickly differentiate into specific cell types. Now, in terms of their body cavities, protostomes exhibit schizocoelous body cavity developments. And the word schizocoelous is one of those words that sounds like the English language hates you here. Schizocoelous. Schizocoelous. The prefix schizo means split, like schizophrenia. If you have schizocoelous body cavity developments, what that means is that a mass of mesoderm splits away from the rest of the mesoderm and the coelom forms within it. And finally, the initial opening into the body, the blastopore. You might remember the blastopore, that primal opening that first forms. If you are a protostome, the blastopore is going to become your mouth. The blastopore becomes the proximal opening to the digestive tract. So protostomes feature spiral cleavage, determinate cleavage, schizocoelous body cavity developments, and the blastopore becomes the mouth. Examples of protostome animals will include, among other things, flatworms, nematodes, that is roundworms, mollusks, annelids, annelids, that is going to be a roundworm or segmented worm, not the same thing as a roundworm, my apologies. Arthropods, arthropods, your insects, crustaceans, and so forth. And a couple of lesser known groups of animals like nemerteans, Nemerteans, which are called ribbon worms, rather primitive aquatic animal, and a group of animals called rotifers, microscopic animals, rotifers. So these are just some examples of animals that exhibit protostome development. It's not a comprehensive list. Now, how about deuterostomes? Deuterostome developments is going to be characterized by some different features. Rather than spiral cleavage, deuterostomes exhibit radial cleavage. And what that means is that in a deuterostome, new cells are laid down perpendicular to the long axis of the embryo or parallel to the long axis of the embryo. So either way, you're going to have radiating out from the middle. You're laying down new cell layers from the central axis, moving outward, either perpendicular or parallel. That's radial cleavage. You will find that in deuterostomes, rather than determinate cleavage, you have indeterminate cleavage. In indeterminate cleavage, that means that differentiation occurs later in embryonic development, that you do not have an early assumption of specific cellular identity by embryonic stem cells. Determinate cleavage, that specific cellular identity forming early on, means that the organism is pretty locked into its configuration early in life. Indeterminate cleavage means that embryonic stem cells can still break away from the main embryo until they differentiate and become new embryos. So indeterminate cleavage makes the possibility of an identical twin. If we exhibit indeterminate cleavage, we are deuterostomes. And what happens when you have an identical twin is that early in the development of that cellular body, that ball of cells, some of the embryonic stem cells break away and they form a new ball of cells, which then goes on to develop into a whole new individual, genetically identical to the ball of cells that it came from, because they came from the same initial fertilization event. So that's how you get identical twins or identical multiples of any kind. You have to have indeterminate cleavage for that, because determinate cleavage, the cells assume specific identities early on and thereafter cannot form a whole new organism after they break away. You will find that rather than schizocoelous body cavity developments, my deuterostomes exhibit enterocoelous body cavity developments. Enterocoelous. And I see that is also misspelled on my handout, spelled L-U-S, not L-O-U-S. I need to go back and make some corrections here. But in enterocoelous body cavity developments, rather than masses of mesoderm splitting away to form the coelom, you're going to find that the coelom forms within the mesoderm and buds from its wall into the other layers, from out pockets, and buds outward into the wall of the archenteron, the primitive digestive tube from the endoderm. And finally, in your deuterostomes, the blastopore, that primal opening in the organism, becomes the anus rather than the mouth. That's, in other words, the first opening becomes the distal end of the digestive tract rather than the proximal end of the digestive tract. So anytime you are feeling like, you know, you're on top of the world, you're better than everybody else, don't forget, all of us started out as an asshole. All right. Now, animals that fall under the heading of deuterostomes. There are not a lot of these as compared to other things. The only ones you are probably well familiar with are going to be echinoderms, that is sea stars, sand dollars, and their affiliates, and chordates, C-H-O-R-D-A-T-E, members of phylum Chordata, our own phylum, including all vertebrates. There are a few other weird phyla of life that are deuterostomes, brachiopods, bryozoans, phoronids. You've probably never heard of any of these things, but those are going to exhibit deuterostome developments. So you're always either protostome or deuterostome. We, as a chordate, are deuterostomes. All right. Now that's my initial look at body plans. We're going to be saying more about them as time goes on. My next lesson is going to involve a closer look at animal anatomy and physiology. And indeed, much of the rest of this course is going to focus on looking at specific organ systems in animals and learning more about the specific developments of each organ system within the body plan, and therefore how they vary from one animal to another and how they help those animals adapt to their environments. But these concerns, these very basic concerns of putting the body plan together, things like developmental pattern, whether or not you have a body cavity and where it comes from, whether or not you exhibit symmetry and what form of symmetry, and how many primary germ layers you have when you're giving rise to your tissues. All of these are fundamental concerns in how the body plan is made to begin with. So I kind of wanted to put them up front. Now, to finish out this little chapter on the basics of Animalia, let's talk for a minute about animal systematics. Let's talk about the classification of animals. Now, you might recall, if you listened to my lesson on plants, that plant taxonomy is a mess, that there is a lot of argument over the classification of plants, that there are a lot of plants that have been given more than one name in different parts of the world, that there is a lot of argument over how many phyla of plants there even are, and whether or not, say, the phylum Anthophyta should be split into multiple phyla, how closely algae are related to plants and whether they should be considered plants or not. You know, these are all basic concerns in plants classification. Animal classification is equally problematic. There are a lot of borderline issues looking at very simple organisms called protozoa, that is, animal-like protists, and asking ourselves the question, okay, is this fairly complex protist really an animal? Does it belong in the kingdom Animalia? Or is it just a complicated colonial protist? We also have a lot of disagreements on how to classify and sort out the kingdom Animalia. There has been a lot of argument over this over the course of the history of biology. We have reclassified many groups of animals on many occasions as we've learned more. Some of this has involved finding fossils that taught us things we didn't know previously and having to say, oops, this belongs better over here than over there. Some of this has involved us discovering new kinds of animals and trying to figure out where they fit in. Some of this has involved very simplified early classifications made by people in the early days of taxonomy that we realized later were much more complex than they thought. And a lot of this in recent times is genetic, looking at the DNA sequences of animals and therefore having to rewrite our phylogenies on the basis of what the DNA tells us about who is more closely related to who. There is still room for dispute in some of these. In a lot of cases, the absence of a known common ancestor makes it hard to sort out classification groups. There are a number of competing hypotheses for how to split up the Kingdom Animalia, and indeed the version I'm giving you right now is revised from the version I would have been giving you if I were teaching this same lesson 6 months ago. But what I'm going to give you right now is a broadly accepted animal taxonomy. You will find many zoologists who disagree with what I'm about to say, but this is the most, you might say, standard division of animals that we have at the present time. So, all animals, all living animals, fall into one single monophyletic clade. That is, all living animals have a single common animal ancestor. That singular group is called the metazoa, capital M-E-T-A-Z-O-A. The word metazoa encompasses all living animals. In fact, you could arguably use the words animal and metazoan synonymously. And if you're looking for a formal name for the Kingdom Animalia, if you called it the Kingdom Metazoa, you wouldn't be wrong. But all living animals fall within the metazoa. We can divide the metazoa into two broad groups. The parazoa, capital P-A-R-A-Z-O-A, and the eumetazoa, capital E-U-M-E-T-A-Z-O-A. The parazoa consists of the most primitive living animals, the sponges. Parazoans represent the oldest branch of the Kingdom Animalia, and those with the simplest body plans. Parazoans are going to, in the modern, there were more parazoans in the past. The parazoa today include exactly one phylum of life, one living animal phylum, the phylum Porifera, capital P-O-R-I-F-E-R-A, the phylum of the sponges. So all living parazoans are sponges. By the way, I didn't say this at the beginning, but I should have. Animals are much more phyletically diverse than any other group of life we have identified to date. You might recall the plant kingdom had 10 living phyla within it. The fungus kingdom has 5 living phyla. The animal kingdom, as presently constructed, has 35 living phyla of animals. We are not going to talk about all 35 of them. We do not have time for that. But I'm going to give you some important examples as we go along, and they're going to be the focus of a lot of the lab activities if you're in the class that I'm in. So, metazoa, broad group of all animals, Kingdom Animalia is the same thing, 35 living phyla. Two large groupings within the metazoa, the parazoa, which today consists only of the phylum Porifera, the sponges, and the eumetazoa, which consists of the other 34 living phyla of animals. Now, within the eumetazoa, 34 living phyla of animals, there are a couple of phyla that are sort of doing their own thing. We have the phylum Placozoa, capital P-L-A-C-O-Z-O-A, which literally translates as flat animal. Placozoans are flat-bodied, very primitive animals. They're about the simplest animals that have true tissues, only a step above sponges in complexity. Seabed dwellers, you don't really see a lot of them. They're the simplest eumetazoans. Also under the eumetazoa, we have the phylum Cnidaria, that is the jellies, corals, anemones, and hydras, characterized by their simple sac-shaped bodies. We have the phylum Ctenophora, C-T-E-N-O-P-H-O-R-A, which are called the comb jellies. They're another variety of jelly. At one time, we thought that the Cnidaria and the Ctenophora had a close evolutionary relationship. We have determined that they are not particularly closely related. There's not one common jelly ancestor. So there are those three distinct phyla within the eumetazoa that are not closely related to other eumetazoans nor to one another. And then there's a single larger group of eumetazoans, the group known as the Bilateria, capital B-I-L-A-T-E-R-I-A. Well, if I didn't say so before, the distinctive feature of the eumetazoa, the thing that sets them apart from the parazoa, is eumetazoans have true tissues. Parazoans do not, eumetazoans do. So within the eumetazoa, we have the Ctenophora, the comb jellies, the Cnidaria, jellies, corals, anemones, and hydras. We have the Placozoa, the flat-bodied basal eumetazoans. And finally, we have the Bilateria, which you can probably guess from the name, consists of all bilateral animals. The Bilateria are a monophyletic group. All bilaterians have a common bilaterian ancestor. There are at present 31 identified phyla within the Bilateria. Now we can divide the Bilateria up a little bit further. We have the phylum Xenocoelomorpha. There's a fun spelling for you. Capital Xenaceelomorpha. Xenocoelomorpha. Don't say xeno, it's an easy trap to fall into. Xenocoelomorpha are the very simplest bilaterians, the most basic bilateral animals. They are a bilateral animal that lacks a true distinct gut. They lack a true digestive tube. Only bilaterians that are like that. Then we have the Protostomia, capital Protostomia, which, as you can probably guess from the name, is all protostomes. They are not the only protostomes. Pretty much everything else we've talked about previously is a protostome, but this is a group that consists exclusively of protostomes. We divide the Protostomia up further. There are 27 phyla recognized at present within the Protostomia. 9 of those phyla fall into a group called the Ecdysozoa, capital Ecdysozoa, the Ecdysozoa, 9 living phyla of ecdysozoans. Now the superphylum Ecdysozoa, superphylum, a big group of phyla, or the ecdysozoans, at present consists of 9 living phyla. I'm not going to go through all of them, but some example phyla of ecdysozoans that you probably know about would include the phylum Arthropoda, capital Arthropoda, the arthropods, your crustaceans, your arachnids, your insects, all of these are arthropods. Also, the phylum Nematoda, capital Nematoda, phylum Nematoda. Your roundworms, a lot of our parasitic worms, like tapeworms, are examples of nematodes. Leeches are nematodes as well. And finally, we have the phylum Tardigrada, capital Tardigrada, the tardigrades. You've already heard of tardigrades, little microscopic animals. They're sometimes called water bears, and they are basically indestructible. Tardigrades can live through just about anything. We have found tardigrades that were accidentally placed onto spacecraft. They ended up accidentally on spacecraft that we launched into orbit, and they were still alive when they got back down to Earth. Tardigrades are some of the hardiest organisms in existence. So there's just a couple examples of phyla within the Ecdysozoa. Also within the Protostomia, we have another superphylum called the Spiralia, capital Spiralia. Superphylum Spiralia consists of all of those phyla that exhibit spiral cleavage during their development. There are 18 living phyla within the Spiralia. Some example phyla that you might be familiar with. We have the phylum Mollusca, capital Mollusca, the mollusks. That consists of most of your critters that have a seashell. That consists of our slugs. That consists of our snails. And that consists of the cephalopods. Your squishy tentacled critters like the squid and the octopus are all mollusks. Also in this group, we have the phylum Annelida, capital Annelida, the annelids, which are your segmented worms, like the earthworm. You might have noticed a lot of different kinds of worm phyla. Worm is a pretty basic and simple invertebrate body plan, and not everything called a worm belongs to the same biological group. There are about 7 different phyla that we call worms. And so the annelids are the true worms, you sometimes hear them called, or the segmented worms. And then also within this group, we have a phylum I really like called the phylum Platyhelminthes, capital Platyhelminthes. Phylum Platyhelminthes consists of flatworms. A lot of parasitic worms are going to be flatworms, but also some very simple and harmless ones. My favorite example being an organism some of you may have played with in high school biology called planaria, capital P-L-A-R, sorry, a P-L-A-N-A-R-I-A, planaria or planarian worms. Planaria are freshwater flatworms. They are characterized by their flat bodies with no internal cavities. Planaria are highly regenerative. You can cut a planarian in half, and chances are both halves will grow into a new flatworm. So they can reproduce via fragmentation as well as sexual reproduction. These are just some examples of the 18 phyla that we find within the Spiralia. And last but not least, within the bilaterians, we have the Deuterostomia, capital D-E-U-T-E-R-O-S-T-O-M-I-A. The Deuterostomia, as you probably guessed from the name, is unified by the quality of being deuterostomes. They exhibit deuterostome developments. The term protostomia for the Ecdysozoa and Spiralia is a little misleading, I think, because it suggests that everything that's a protostome falls within those groups. Everything other than the deuterostomia exhibits protostome developments. So just because 27 of those phyla are called the protostomia does not mean that, you know, cnidarians and xenocoelomorphs are deuterostomes. They are protostomes too. But the Deuterostomia, that group, consists of 3 living phyla. There is the phylum Hemichordata, capital Hemichordata, the hemichordates. Common name for them is the acorn worms. You've probably never seen one. They're seabed-dwelling worms that dig burrows in the silt on the bottom of the ocean floor. We've also got the phylum Echinodermata, which I've mentioned a few times already in this lesson, capital E-C-H-I-N-O-D-E-R-M-A-T-A, the echinoderms, largely consisting of your sea stars and your sand dollars and their affiliates. And finally, the phylum Chordata, capital C-H-O-R-D-A-T-A, the chordates, which includes all organisms that have a dorsal hollow nerve cord running down the back length of the body, a notochord, N-O-T-O-C-H-O-R-D, which is a flexible supportive structure that the spine is built around if you happen to have a spine, and also a post-anal tail. That is a tail that extends past the animal's anus. Chordates all are organisms that have those features. The phylum Chordata contains the subphylum Vertebrata, capital V-E-R-T-E-B-R-A-T-A, Vertebrata, or the vertebrates. We, of course, fall within the subphylum Vertebrata, which includes all reptiles, all birds, although you heard me say last time, let's lump them together as the Sauropodomorpha, all amphibians, all sharks, and, of course, all mammals. There are some chordates that are not vertebrates. For example, we've got a little animal called the sea squirt and the sea cucumber. These are both examples of chordates that are not vertebrates. But the phylum Chordata is where we belong. So that's our overarching animal phylogenetic breakdown. Metazoa for everything, divided into parazoa containing only the porifera, phylum Porifera, and the eumetazoa, all animals with true tissues. Eumetazoa contains the Ctenophora, or comb jellies, the Placozoa, or basal flat-bodied animals, the Cnidaria, your jellies, corals, and anemones, and the larger group called the Bilateria, which consists of 31 other phyla. Within the Bilateria, we have the phylum Xenocoelomorpha, the simple basal bilaterians with no true gut. We have the Protostomia, which consists of the superphylum Ecdysozoa, that is all those organisms with an exoskeleton, and the superphylum Spiralia, all those organisms with spiral cleavage, 27 phyla between those two groups. And finally, the Deuterostomia, consisting of 3 phyla, including our own phylum, the Chordata. That is the presently recognized systematics of the Kingdom Animalia. Again, there's a lot of argument about which specific phyla belong where and which larger supergroupings are valid groupings. This is the best we've got right now. This is the most widely accepted division of the Kingdom Animalia at present. There are zoologists who would disagree with me about some of these classifications, but it is the most standard, if you will, division of the Kingdom Animalia with our present understanding. 35 living phyla within this, quite a few non-living phyla. There are a lot of extinct animal phyla that we only know about from the fossil record, and no doubt there are numerous extinct phyla that we have no evidence of at all because they didn't leave a good fossil trace behind. All right, so having finished up our look at animal systematics, we are now done looking at the fundamentals of Kingdom Animalia. My subsequent lessons in this chapter are going to deal, well, not this chapter, in this unit, are going to deal primarily with animal anatomy and physiology, taking a look at animal body parts and specific organ systems, answering basic questions like how do animals get their energy, how do animals obtain oxygen, how do animals process their food and circulate nutrients, all those kinds of things. We're going to talk about several distinct animal organ systems, and along the way we're going to talk about numerous representative phyla of animals. The ones that I've named in our discussion today, primarily the phyla that you see within the Bilateria, are going to get special attention from us. They're going to be phyla that I'm going to mention at some point specifically because they have particular interesting features that help us to illustrate a lot of what is going on in animal adaptation and development. All right, so that is it for this particular group of lectures. I hope you've enjoyed. Thank you for your time. I'll talk to you all later. Good night.